Let's start by understanding systemic and opportunistic mycoses. These are fungal infections that either affect the whole body or take advantage of weakened immune systems. The key thing to remember is that systemic mycoses often involve deep tissue or organs, while opportunistic mycoses happen mainly in people with compromised defenses. This sets the stage for exploring how these fungi interact with the human body and cause disease. Now, systemic mycoses are infections caused by fungi that typically come from the environment, especially soil. They include diseases like coccidioidomycosis, histoplasmosis, blastomycosis, paracoccidioidomycosis, and cryptococcosis. Each is caused by a specific fungus inhaled from soil particles. What's important here is that these infections can affect multiple organs, sometimes including the central nervous system, especially in the case of cryptococcosis. Understanding the source and nature of these fungi helps us grasp how systemic infections develop. So, what exactly are opportunistic mycoses? These occur when fungi that usually don't cause harm take advantage of weakened immune systems. We often call this the disease of the diseased, meaning it mostly affects the very young, the very old, or those with serious illnesses. Groups at risk include people with HIV, organ transplant recipients on immunosuppressants, cancer patients undergoing therapy, and those with diabetes. This helps us see why immune status is crucial in fungal infections. When we talk about opportunistic fungal infections, the main culprits fall into two groups: yeast-like fungi, such as Cryptococcus and Candida, and filamentous fungi, like Aspergillus, Mucor, Absidia, Rhizopus, Fusarium, and Penicillium. These fungi are usually harmless but can cause serious infections in vulnerable patients. Knowing these causative agents helps us target diagnosis and treatment effectively. Candidiasis, also known as moniliasis, is a common fungal infection caused mainly by Candida albicans. It can appear superficially, affecting the skin or mucosal membranes, or systemically, affecting internal organs. Besides C. albicans, other species like C. tropicalis and C. cruzi can also cause infection. These fungi grow as oval budding yeast cells and sometimes form pseudohyphae, which helps them invade tissues. Recognizing these growth forms is key for diagnosis. Superficial candidiasis manifests in various ways. It can cause mucocutaneous lesions like oral thrush, vulvovaginitis, balanitis, and eye infections such as conjunctivitis and keratitis. Skin and nail infections, including onychomycosis, are also common. In infants, a typical sign is napkin dermatitis, a diaper rash caused by Candida. These manifestations highlight how candidiasis affects different body areas, often linked to moisture and immune status. Now, oral candidiasis usually comes from fungi already present in the body. Candida's ability to stick to tissues and dental prosthesis like dentures allows it to form biofilms. This helps the fungus survive and invade deeper tissues by forming hyphae. Cell-mediated immunity plays a crucial role in defending against this infection. Clinically, oral candidiasis appears in forms like pseudomembranous, erythematous, and hyperplastic, each with distinct features and severity. Comparing the types of oral candidiasis helps us understand their differences. Pseudomembranous candidiasis, or thrush, shows white curd-like membranes that can spread easily. Erythematous candidiasis presents as red, smooth patches, often without symptoms, but linked to risk factors. Hyperplastic candidiasis, also called chronic leukoplakia, features raised white plaques and carries a risk of malignant transformation. Knowing these distinctions guides diagnosis and treatment decisions. Looking at clinical images of candidiasis reinforces what we've discussed. Severe oral thrush shows thick white plaques covering the throat, while angular colitis causes cracking at the mouth corners. Diaper rash in infants appears as bright red, inflamed skin, and candidal paronychia affects the nail folds with swelling and discoloration. These examples illustrate how candidiasis can affect various body parts with distinctive signs. Systemic candidiasis involves more serious infections beyond the skin and mucosal membranes. It can affect the intestines, lungs, bloodstream, heart, brain, urinary tract, and kidneys. This wide range shows how Candida can invade multiple organs, especially in immunocompromised patients. Recognizing these systemic forms is critical because they often require aggressive treatment and can be life-threatening. Diagnosing fungal infections depends on collecting samples from the affected site. Common methods include microscopic exams like wet mounts, gram staining, and KOH mounts. The germ tube test helps identify Candida species. Slide culture techniques reveal chlamydospores, and cultures on suburodextrose agar confirm fungal growth. Molecular tests like PCR provide precise identification. Combining these methods improves diagnostic accuracy. Diagnostic images give us a closer look at fungal identification. Microscopic views show Candida hyphae, highlighting their structure. Gram stains reveal budding yeast cells and pseudohyphae, characteristic of Candida. Slide cultures demonstrate how fungi grow on media, and Petri dish cultures display creamy white Candida albicans colonies. These visuals are essential for confirming infection and guiding treatment. Comparing antifungal agents helps us understand how different drugs work. Polyenes like nystatin and amphotericin bind to ergosterol in fungal membranes, altering permeability. Azoles interfere with ergosterol synthesis, disrupting the cell membrane. DNA analogs like flucicide inhibit fungal DNA and RNA synthesis. Echinocandins disrupt the fungal cell wall integrity. Knowing these modes of action assist in selecting effective treatments based on the infection type. Cryptococcosis is caused by Cryptococcus neoformans and can be both opportunistic and systemic. It mainly affects immunocompromised individuals and often leads to cryptococcal meningitis. Diagnosis involves microscopy using India ink staining, gram staining, and culture methods. Antifungal treatment is essential for managing this serious infection. Understanding these points helps in early recognition and intervention. Microscopic images of Cryptococcus neoformans reveal encapsulated yeast cells clearly visible against a dark background. Stain samples show these cells as gram-positive budding yeast cells. Aspergillosis is caused by species like Aspergillus fumigus, flavus, and niger, which are common in the environment. Clinical syndromes range from allergic reactions to invasive diseases, particularly in immunocompromised patients. This infection can become disseminated, affecting multiple organs. Invasive aspergillosis and chronic necrotizing Aspergillus pneumonia are serious forms. Recognizing these patterns is vital for timely diagnosis and treatment. Zygomycosis, also called mucormycosis, involves fungi like mucor, rhizopus, and absidia, which are widespread in the environment and usually saprophytic. Risk factors include diabetes, blood cancers, corticosteroid use, severe burns, and immunodeficiencies. This diversity in presentation requires awareness for early detection and management. Looking at fungal structures, we see septate hyphae with nuclei separated by septa and aseptate hyphae lacking these divisions. This difference is important for distinguishing fungal types. The Petri dish images show various Aspergillus species, each with unique colony appearances, from fluffy white to darker colors. These visual clues help in identifying species, which is crucial for diagnosis and treatment planning.